So we're going to do animals for the rest of the semester. We're going to do a combination of sections, different features, functions of animals, and we'll devote a little bit of time to the different main animal groups so that when I talk about this happens in this group and that happens in that group, you'll have some understanding of how the groups work. Piled hyper and deeper gives you a good idea of what it's like being in graduate school. In the first box it goes, through my genius, I will transform this lifeless mass into a being of pure academic rigor. They will tremble before my name. Second one, arrogant they called me, drunk on illusions of grandeur and godhood. I said, duh, I'm a professor, of course I think I'm a god. As of course he's not. These are graduate students that he's forming into beings of pure academic rigor. Here's the termite queen and her egg chamber. We're going to talk about reproduction. Reproduction will give us some terms that will then apply to a lot of the animal groups when we get to the animal groups. So this chapter is on animal reproduction and development. It's chapter 11. Every generation needs to be able to produce the next generation. And what you find in animals, this applies to plants too, it applies to living things pretty much across the board, is that what's the typical number of offspring in this species or that species or this other species? And it's associated with the survivability. What are the odds that this offspring will live long enough to produce its own offspring? If the odds are pretty bad, you better crank out an awful lot of them. If the odds are pretty good, you don't need to produce huge numbers of offspring. You think our relatives, the apes, we're pretty good at raising and protecting our offspring. We're pretty decent at getting them to an age where they can reproduce. And so we don't produce that many of them. Whereas an insect, where its offspring are going to be picked off by all sorts of different predators, it better crank out a huge number of offspring on average, or the next generation isn't going to make it to be the next generation. So that's it's an inverse, what they call inverse relationship. The lower the survivability, the more offspring are typically produced. This is one of the problems you wind up with in, sometimes we look at third world countries and we go, you guys are making way too many babies. Is that this population explosion is not sustainable. But we don't take into account that those babies don't have the odds of survival that our babies have. And if in a situation where conditions are getting better, you're gonna tend toward more offspring than you should, because it takes a while to compensate. It takes a while for the birth rate to kind of come down to match the survivability. It did in our country, it does in other countries. It does eventually do that. Some of this stuff we will have talked about before. Asexual reproduction can involve splitting in half, binary fission. That's unusual in multicelled animals. It can involve budding, where a small offspring grows off of a larger adult. It may involve fragmentation, where an adult can break itself into pieces and each piece can generate into a new animal. Things like sponges can do something like that. To some extent, some cnidarians, some corals can do stuff like that. Starfish can break an arm off as a defensive measure, and in theory, that arm can generate a whole new starfish and it can regenerate the lost arm. It's not actually a form of reproduction, even though it can reproduce that way. So it's sort of fragmentation, but it's not considered fragmentation. It's considered regeneration of the missing pieces. Now, we've talked about sexual reproduction. I mentioned that the approach of male-female is extremely common in animals, so we've talked about the differences between male and female. All comes down to what sort of sex cells you make. Those sex cells are called gametes. Sperm and ova are gametes. We talked about how production of egg cells involves a starting cell producing one egg cell and 3 polar bodies. The timing of that production changes depending on the animals. Some animals, when the sperm meets the egg cell, it isn't really an egg cell yet. That's true in humans. Is our second meiotic division does not happen until a sperm nucleus gets into the egg cell, which is technically a secondary oocyte. And then it does, it's already made one polar body during ovulation. Now it's got to make the second one before it can merge the two haploid, one set of chromosome nuclei together in order to produce the next generation. Testes and ovaries is pretty much almost a universal term in males and females in animals. Why in mammals we call them testicles? They are testes, but we call them testicles. I don't know why. And of course the testes produce the sperm and the ovaries produce the egg cells. You have what are called primary sex organs and secondary sex organs. Primary sex organs are the ones that are involved in making the cells, and the secondary ones do the other things that you need in order to reproduce. They may make the hormones. They may involve copulation success. And then you have secondary sex organs. And of course, in female systems, they may, the accessory organs may involve packing the eggs, packaging the eggs, packing the offspring inside eggs or whatever it is that you're packing in order to get your offspring out and about in the world. There are features that are primary sexual characteristics. Those are absolutely necessary for successful reproduction. And then there's secondary sexual characteristics. In people, having a working penis is a primary sexual characteristic. Having working testes and semen glands is a primary sexual characteristic. But beards in males is a secondary sexual characteristic. The tendency when we grow through our teens for our shoulders to grow broader, the tendency for us to put on muscle bulk easier than females do is a secondary sexual characteristic. Breasts, which is a very mysterious sexual characteristic in people because other mammals don't build and sustain breasts through their whole lifetime. They build them when they're going to need them to nurse babies, and then they kind of disappear. But in female, breasts are secondary sexual characteristic. In female humans, a fat layer under the skin is sort of in between. It's considered a secondary sexual characteristic, but those fat cells are actually a backup source of estrogen, and if you don't have enough fat cells, you may not make enough estrogen for the system to work. So it's sort of maybe primary, secondary. But secondary sexual characteristics are, a peacock tail is a secondary sexual characteristic. Doesn't absolutely need that tail in order to make babies, but it kind of needs that tail in order to get the females to let them make babies. So, but it's secondary. We've talked about dioecious and monoecious. Two separate sexes and separate individuals versus two sexes in the same individuals. And in a lot of animal groups, that is a characteristic of either the entire phylum or subgroups within the phylum, where a group will be monoecious, a group will typically be dioecious. And it's biology, so you'll find exceptions. the old term is hermaphroditic for monoecious. Whether, and this is true for plants too, whether an animal can be monoecious doesn't necessarily mean it can self-fertilize, doesn't mean it can make babies by itself. Is that there's a number of things that get in the way. Sometimes it's just not physically possible, and in some cases, the combination genetically will be, I can't think of the word, but it won't work. In some animals, you find parthenogenesis, where females can generate an embryo using an unfertilized egg. That turns out to be somewhat unusual. And, you know, from a genetic and adaptive standpoint, it should have some pretty limiting, limiting variability, and so limiting potential as an evolutionary approach. But you do find it in reptiles and fish and some other things. And we talked about alternation of generations in the plants. We're going to wind up with alternation of generations in animals as well. Budding. He looks just like you, but he looks just like me too. Asexual reproduction. I will write some of these things down eventually. Doesn't work exactly this way in all animals, but it gives you an idea. Sperm and the egg cell, two haploid cells, one set of chromosomes, get together. Let's see. Sperm and egg cell. They eventually fuse their nuclei together and produce a nucleus with two sets of chromosomes. That is now this is fertilization. Fertilization, and it forms a zygote. Zygote is the first cell that's going to be the offspring. And the sperm and eggs are produced by meiosis, reduction division. It gives you one set of chromosomes per cells. But once you get a zygote, it reproduces by mitosis. It makes copies, distributes its genetics in copies to all the new cells. At least that's the way it's supposed to work. And what you typically get, and this will vary from animal group to animal group, but in animal groups like ours and a number of other things, you get either a solid ball of cells that hollows out, or a layer of cells around a food source that will eventually pucker in and it will pucker in, and the outside cells will flow to the inside and form an inner layer, again, paralleling the outer layer. Now you've got an outer layer and an inner layer. Ectoderm, outer skin, endoderm, inner skin. And those are gonna have different fates. They're going to be different parts of the final animal. And eventually you'll get a layer in between called mesoderm that's going to form even more different things. So, time to write some stuff down. The egg needs a way to let one sperm nucleus in and shut the other ones out. It can deactivate the activities of the enzymes the sperm are using to get through the membrane. That doesn't always work. Sometimes you do get two or more sperm nuclei into the egg cell, and then it's a matter of what's the response. In some, they're immediately moved to the membrane and push back out everything but one. In some, there are things inside the cells that all but one is actually attacked and broken down like it was food. In some, they crank out lots and lots and lots of offspring. They let all of the nuclei fuse. You wind up with a triploid or a quadruploid zygote, it won't last very long. Way too many copies of all those genes. But you've got so many other potential offspring that you can, why do anything about it? Let it die. But diploid zygote, cell divides and divides and divides and divides. That's called cleavage. Oh, I got to mention something. I keep saying the sperm nucleus gets in. That is all that should get in. A sperm is a mobile cell. It's got a mitochondria in it. It's got a little organelle that is an energy source for swimming tail or however the sperm is moving. Mitochondria were endosymbionts. They started as ingested bacteria with really useful features, functions, and over the course of billions of years, they've become an integral part of the cells. But they do still have their own little bacterial genes in them, which means that potentially there could be a compatibility issue between the mitochondria of the sperm from the father and the mitochondria of the mother from her egg cell. Doesn't always matter, but in a lot of animals, especially where males and females are separate, because all your mitochondria should be your mother's mitochondria. They should all be genetically identical, more or less, except for mutational differences. But different people, you don't want to have two mitochondria in the same zygote. They don't necessarily get along. As I said, your mitochondria came from your mother. It turns out every once in a while somebody's mitochondria came from their father. The sperm mitochondria got into the egg, didn't get along with the egg's mitochondria. Whatever the conflict was, the egg cell mitochondria went away, and so only the paternal mitochondria are there. Is that it took a long time for them to discover this, exceptions. Where does fertilization occur? In a lot of aquatic animals, the sperm swim and the egg cells are released into the environment and the sperm are deposited near them or released near them. And the sperm find the egg cells and that's external fertilization. In some animals, the sperm have to be introduced into the female, and that's where the sperm will swim through tissue fluid of one sort or another to get to the egg cell. And land animals, that's very common. And it's also common in some aquatic animals where it's better for the eggs to be protected inside the female's body, or where fertilization happens and where the offspring are going to be deposited in very different places. So the female needs to move to where she's going to deposit the offspring eventually. There's a number of different things for internal fertilization. It isn't always an aquatic and land dichotomy. Is that there's a lot of aquatic animals, especially arthropods, that use internal fertilization. The pattern in which the cells divide is called cleavage. It's not unusual for the zygote itself and the cluster of cells to have already an asymmetry where on one end some part of the animal is going to form, the other end another part of the animal is going to form. That's called polarity. They didn't used to think that mammals had polarity, but a lot of study into in vitro fertilization and such has found that our egg cells do have a certain amount of polarity, and where you introduce the sperm to an egg cell when you're doing it artificially has a lot to do with the success of the fertilization and development. Oops. I'll explain this in a minute. Sometimes if I say too much while I'm writing, I either write the wrong thing or I say something I didn't mean to say. There's two different ways that cells divide, that divide two major trunks of animals, the deuterostomes and the protostomes, and we'll talk about other differences eventually. Radial cleavage, the cells divide, but they don't divide evenly. You get a large cell and a small cell on the divisions. And the small cells migrate in a way where the outside of the developing embryo looks like it's got a spiral of little cells on it. So that's called spiral cleavage. Radial cleavage, if you're looking down at the division, you get a division and then a division and then divisions. And you see radii, a bunch of more than one radius. And that's radial cleavage, and the division is relatively equal. Radial, well, let me go down here. Spiral cleavage, very, very early on, as those cells divide, cell number 32 already is almost completely locked into what it's eventually going to be in the animal. Whereas for a very, very long time in radial animals like us, the cells are very flexible, is that they're not locked into a particular fate down the road, and that allows us to have human stem cells and other kinds of things that are fairly useful, and to produce identical twins where an embryo divides into two pieces, and each one of them can develop into an offspring. In a spiral cleavage, a protostome, you divide the embryo into two pieces. Part of it was going to be the front, part of it was going to be the back, or something like that. It doesn't really form two complete embryos. Now the name, protostomes and deuterostomes, the protostomes, that dent that's going to flow in and form the endoderm, that's an opening on the outside of the hollow ball. We'll get that name up there in a second. And that opening is going to be the mouth proto in one group, and it's going to be theoretically the anus in the other group. It actually most of the time forms the opening of the umbilical cord, but theoretically it's the anus. And that internal layer is, not too surprisingly, going to be mostly digestive system. We'll get to that. The blastula is the hollow ball of cells. When it dents and you get layers, it's called the gastrula. That's called gastrulation. And you get 3 different germ layers. I'm just going to squeeze this in here, because this is all we're going to do for this particular video. So leave yourself room to write. So the endoderm, the inner layer, it's going to be the digestive system and things associated with the digestive system. In us, our lungs grow off of the digestive system. Very commonly, gills grow off of the digestive system. There are a few other things, part of your liver and part of your pituitary gland. But a whole bunch of things grow off of this tube inside the body that's going to be the endoderm. The ectoderm, what you would expect, the outer structures, the skin. If there's an outer skeleton, if there's an exoskeleton, the exoskeleton is ectodermally derived. And this one's a little bit of a surprise. The nervous system. Nervous system forms on the surface and then sinks into the animal, or is pulled into the animal when it pulls the skin up on both sides. And things associated with the nervous system are ectoderm. And then mesoderm's kind of pretty much everything else, a whole bunch of internal organs, your circulation system, your waste removal system, your muscle system. If you have an internal skeleton, your internal skeletal system, a lot of glands and other things are part of mesoderm. And there's a lot of crossover if you're looking at particular systems, where, yeah, your digestive system is mostly endoderm, your nervous system is mostly ectoderm. Your gland system is actually a combination of a bunch of them. Okay. What will eventually happen, I also should mention that let's see. One other difference. The protostomes, the protostomes form mesoderm here, and it fills the body. The deuterostomes have little outpocketings along the endoderm that expand and form the mesoderm. So there is a difference between how protostomes and deuterostomes form mesoderm as well. Once the layers are in place, then you get the cells start to dip into different genetic packages and do what's called differentiation. They took the word different and made, a bigger word out of it. And then you get the tissues and the other sorts of things we're going to talk about farther on in this chapter.